Hi everyone, my name is Brayan Vanegas. I study at the National University of Colombia. Here I will be discussing our project research titled In Silico Identification of Genome-Wide Short Tandem Repeats STRs markers associated with domestication traits in Lima bean Phaseolus lunatus. As a brief introduction, lima bean is a legume widely consumed worldwide that experienced gradual domestication, once in Mesoamerica and once in the Andes, which were characterized by a strong founded effect. Recently, interest has increased in deciphering the genetic basis of domestication and agronomic traits in this species. Population genomics studies in lima bean have mainly focused on single nucleotide polymorphism, leaving behind other types of genetic variations, such as short tandem repeats, that could have contributed to adaptation of lima bean during domestication. The recent release of a high-quality genome assembly of a Colombian domesticated variety of lima bean and the availability of whole genome resequencing data provide a great opportunity to identify genome-wide STR loci associated to domestication and agronomic traits. For this reason, the aim of the present study was to identify in silico a set of genome-wide STR loci informative for population genomics and association studies and to carry out experimental validation. To reach this aim, to identify STR loci, the reference genome of Lima bean published in the year 2021 was used. So first, the DNA sequence of each one of the 11 Lima bean chromosomes was analyzed separately in search for STR loci with the use of the computer program GMATA. The search for STR loci with repeat motif from G to deca nucleotide with a minimum repeat number of 5 was performed in this program. Then, to identify polymorphic STR loci in silico, whole genome resequencing data previously produced for 60 lima beans sections was used in the program HIP-STR. Genotyping data were stored in a BCF file, which was used for genetic diversity analysis. Furthermore, the BCF file was filtered to keep STR loci with high quality genotyping with low percentage of missing data, less than 20%, and minor allele frequency of 5%. Genetic diversity analysis was performed by calculating expected heterozygosity for each of four populations, wild and domesticated sections within the Mesoamerican and Andean gene pools, and the fixation index FST locus by locus using the AdGenet package in R, filtering autologic with the top 10% highest genetic diversity loss and FST values. Those were called outlier loci. The annotation of the outlier loci was used to identify a set of loci potentially involved in the domestication process or traits of agronomic interest. These loci were called candidate STR loci. Of these, a reduced set of 16 STR loci occurring within genes was selected for experimental validation through PCR amplification and Sanger sequencing in different lima bean sections. Now, as a result, a total of 109,698 genomic microsatellite markers were identified across the entire genome of lima bean, which are located on the 11 chromosomes of the bean and range from D to the K nucleotides, as you can see in the figure 1. This has a genome size of 560 megabase, and the density of the detected markers is 1 STR locus per 5,000 base pairs. Within the STRs, a total of 82,522 STR lossy about 75% corresponding to the nucleotides, as you can see. From the identified STR lossy, a total of 26,278 were genotyped using the HIP program, and among these, 9,436 are informative for the Andean gene pool, and 13,396 are informative for the Mesoamerican gene pool. With this set of polymorphic lossy, genetic diversity indexes were calculated within each gene pool. The results showed a strong reduction of genetic diversity in the domesticated material compared to the wild one: 49% for the Mesoamerican gene pool and 33% for the Andean gene pool. Related to outlier lossy, a set of 142 lossy for the Mesoamerican gene pool and 98 lossy for the Andean gene pool were identified in the top 10%. Of these, 16 lossy were selected associated to domestication and agronomic traits to experimental validation. Thus, in the following figure 4, one of the predicted STRs can be seen in this gene, which has been reported as a transcription factor involved in flowering. Therefore, this trait may be being affected if we compare the Mesoamerican and the Andean gene pool, where changes in STR were evident. And the conclusion is the outlier loss identified in this study will be useful for future genome-wide association studies aiming to unreveal the genetic basis of traits of interest for genetic improvement such as drought tolerance. Thanks.